Good afternoon, class. So today we're going to be doing an online lecture for the eye and vision. And this is an online lab lecture. And for this online lab lecture, it focused a lot on the clinical aspect of vision. Okay, but you still need to know the anatomy of the eye. So make sure you do your lab manual pages 351 to 361, and make sure you do the review sheet exercise 23, and that will help you with labeling the structures of the eye so you know all of that information too. So essentially the eye is very sensitive, so you have 70% of all sensory receptors are in the eye. Each eye has over 1 million nerve fibers, and the eye is quite protected. So the majority of the eye is actually in the socket, in the bony orbit, okay? And then the other part of the eye that you can't see is surrounded, it's cushioned in fat. All right, so you have muscles that help control the movement um, of your eye. So you have internal muscles that are controlled by the autonomic nervous system. So you should get used to that word autonomic by now and know that that means that you do not voluntarily control anything that's dealing with the autonomic nervous system. The autonomic nervous system controls, it sends its own signals. It controls essentially itself involuntarily. All right. And so, example is your pupils constricting when they're exposed to bright light. So essentially, you have the pupils, the opening of the iris. Okay, the iris is what makes your eyes brown, blue, different colors is your iris, and essentially the opening of the iris is the pupil. All right, but the iris is going to be controlled. Essentially, you can control the pupil diameter opening, either make it dilated or constricted by the iris, which is controlled by ciliary muscles. Okay, so those muscles are going to manipulate the iris, which is going to make the pupils constrict if it's bright light or dilate if the light is dim, and all that is to essentially allow accommodation to occur. So accommodation is just to allow your eyes to focus on whatever it is you're trying to view. And again, this is going to be essentially a reflex because remember, reflexes, the majority of them are involuntary, meaning there isn't any thought to it. So, and with this case, you don't look at something and say, I want to focus my eyes on this object. You just look at it and your eyes automatically focus on it. Okay. And then you also have external muscles that control the movement to follow objects. So you have your superior oblique muscle of the eye. You have your trochlea muscle. You also have your superior rectus muscle, lateral rectus muscle, inferior oblique muscle, and inferior rectus muscle. All right. And then when you view close objects, you're gonna use convergence, which is just your eyes moving medially. Now we're gonna look at some clinical aspects of vision. But before we do that, we should mention how light actually travels through the eye. So essentially, when light's gonna enter through the pupil, go through the lens, all right, and then it has to travel all the way to the posterior part of the eye. Okay, so it's gonna pass through. You have different layers in the posterior part of the eye, but we're gonna focus on the retina. So essentially, the retina is gonna be the back of the eye, and it's going to include the pigment cells of the outer pigmented layer and the neurons of the neural layer. All right, so essentially light must pass through, it's going to be a ganglion cell layer, and then it's going to be a bipolar cell layer to reach and excite the rods and cones. Okay. All right. Then once that light has excited the rods and cones, the rods and cones are going to send the signal back to the bipolar cells, and then the bipolar cells are going to send a signal to the ganglion cells, and then the ganglion axons are going to form the optic nerve, and that's going to send the sensory signal outside, to the brain, out of the eye into the brain, through the optic nerve. So, and within all of that happening, okay, so you have to essentially, if the light reaches the retina correctly and focuses on the retina, that's called focus correctly on the retina. It's called emmetropia. Okay. But if light from an object fails to reach the retina, then you have what's called myopia, which is nearsightedness. So the distant objects are going to appear blurry. All right, and there will be a diagram of this in the next couple of slides. But essentially, it could result from an eyeball being too long. And if the light from the objects focus behind the retina, you have what's called farsighted hyperopia, and then near objects are blurry while distant objects are clear. And essentially, this could result from an eyeball that is too short or from a lazy lens. And this figure essentially sums up what we got through talking about. So with the emmetropic eye, so that's going to, images are going to focus correctly. You see how the light from the images reach the retina. Okay, so they reach the retina properly. It's not in front and it's not behind, so they focus correctly and you have correct vision. All right, so myopic eye, when you're nearsighted, you see how the image, the focal plane is behind the retina. Okay, I'm sorry, not behind the retina. It doesn't reach the retina. Okay, I'm sorry, it doesn't reach the retina. And then you see with the hyperopic eye that the focal plane is behind the retina. Okay, all right. And you see right here the lenses that correct these types of visions. So with someone's nearsighted, if you use a concave lens, okay, that will cause the light to focus properly on the retina. All right, it's no longer in front of the retina. It focuses right where it needs to be. And then if someone is farsighted, use a convex lens, and that'll take the image that was once behind the retina and bring it to where it should be. Focuses on the retina, all right, to correct that farsighted vision. All right, so with astigmatism, so images are blurry. So this can result from light focusing as lines, not points, on the retina due to unequal curvatures of the cornea or lens. And I have like an astigmatism in one of my eyes, so you still can get glasses to correct that issue also. You can have other homeostatic imbalances of the eye. So essentially, your cones, they deal with color. In order for your cones to function at their best, you need to have a lot of light. All right. And then your rods, they deal with essentially allow you to see at night. Okay, that's going to be deal with your ability to see in low light. You need your rods. They specialize in that. All right. So with night blindness, so your rod function is going to be inhibited. All right. So it hinders the ability to see at night. And then color blindness is actually a genetic condition. So essentially, it's on the X chromosome. And just briefly, so males have an XY, females have an XX, they have two X, males have an XY, and essentially, color blindness is on the X chromosome. So it's a recessive trait, and recessive traits have to either have two copies of them in order to be recessive. If you have one normal copy of a trait and one recessive copy, then the trait's going to be normal. Your vision is going to be normal as far as color blindness. But if you have, in this case, since males only have one X chromosome, so color blindness is usually expressed in males. So essentially, since males only have one X chromosome, so when they get that colorblind trait on their X chromosome, it's going to be expressed on them because they don't have another X chromosome like females to a lot of times counteract that recessive colorblind trait. Okay, so it's you usually see it in males as far as colorblind is usually shown in expressed in males, and it's the inability to see certain colors. All right, usually red, green, something like that. All right, due to the lack of one type of cone, partial color blindness. You can also have partial color blindness. All right, then cataracts. As you get older, you see in older people they get cataract surgeries. So that's when the lens become hard and opaque, and the vision becomes hazy and distorted. So it's something that can be removed. So you can have cataract surgery to remove that opaque film that's covering the eyes. Okay, so that is the end of the notes for the eye section. And again, normally you would have done a sheep eye dissection in lab, but you will still watch the sheep eye dissection. It's in the learning units um, on Blackboard, so watch that, and you do have to do a discussion right up on that. But it's still not the same as doing it yourself. But still do the work pages, the exercise sheets in your lab manual. All right, and that will help you as far as learning the anatomy structures of the eye also. All right, thank you.